right. So while we wait for people to come in, it is my pleasure to introduce Professor Elizabeth Kathleen Neumann. How do you pronounce yourself? Neumann or Newman or?
So I actually leave it up to people to see if they know it's German or not. So I say Newman, but also I respond to Neumann just the same.
Okay, so then we keep it with Neumann, I guess. So Professor Neumann is a recent hire at University of California in Davis since July this year. So congratulations to your appointment. Welcome to UC Davis. And you know, that is well earned. She did a lot of work before. She has been a postdoctoral research fellow at Vanderbilt University in Nashville, Tennessee from April 2019 to June 2022, including getting awards as an NIH Toxicology T32 fellow and NADDK F32 fellow. Before she did her PhD in analytical chemistry at the University of Illinois at Urbana-Champaign from August 2015 to 19, where she was a Springborn Fellow and had an NSF grant. And before that, she did a Bachelor in Science in Chemistry at Baylor University in Texas. So what she published, and she has an impact factor of 16 right now, of really highly cited papers published in 2019, mostly from the Vanderbilt University to current, about three-dimensional imaging and ion mobility mass spec, correlating imaging of the human kidney and staph-infected murine kidney with MALDI timsTOF. She established methods and protocols for establishing tissue normalcy across dozens of human patients, collaborated with HubMap consortium members for multimodal imaging of spleen, heart, lymph node, small intestine, colon, and pancreas, developed methods for 10-micron small metabolite ion mobility imaging, and multiple other work. Before that, she was in the Sweetler lab. So Sweetler is one of the senior editors of the Journal of Organic Chemistry. There she also did single-cell MALDI-MS to tens of thousands of single cells, and did also micro mass spectrometry and other things. So with that, she is a clear star, a rising star, and we are very much inclined to welcome you here and collaborate with you in mass spec imaging, and yeah, take it from here.
Awesome. Thanks, Oliver. That was a very kind introduction. I'm hoping that I share the right screen. Can everyone see the full thing? Cool. Yeah. I see some nods.
Yes, it's good.
Cool. So I do multimodal mass spectrometry, and so here are some examples of what mass spectrometry imaging actually looks like. And so what this technology allows us to do is actually visualize the molecules in a variety of different systems. I specialize in biological systems, although I've done some material science as well. So it's a very flexible technology in that way. And so this is an infected murine kidney. This is a whole body mouse pup. This is a non-infected rodent kidney, a rodent brain, and then a human kidney. So for the most of my talk, I'm going to be focusing on what I've done as a postdoc at Vanderbilt, but I'll talk in the end of what I'm actually doing at Davis. So with that, we'll briefly talk into an outline of why multimodal imaging approaches are important, and then talk about different ways of how you can actually do highly multiplex immunofluorescence for labeling different cell types, and how you can complement this with the rich molecular information by multi-mass spectrometry, and how you can actually integrate these two different technologies together so that you can get a better understanding of biological systems. And then I'll actually end on some pictures of my lab at Davis, as well as what we're planning to do in the Neumann lab. And so what I think about most evenings right before I go to bed is, how do you actually understand biological systems? So if we go to the central dogma of biology, DNA is transcribed to RNA and RNA is translated to proteins. And so before anyone in the audience that's a biochemist throws a fit, I know it's way more complex than this simple schematic. But for the sake of discussion and argument, we're going to tack on metabolites at the end. And so we have DNA going to RNA, RNA going to proteins, and proteins producing metabolites, knowing that metabolites actually interact at each of these different phases as well. And so how do we actually understand these different molecular features within a biological system? As an analytical chemist, I really think about primarily what technologies can you use to visualize or quantitate or measure. And so here's like not an exhaustive list, but just to give you an idea of different ways you can actually study DNA or RNA or proteins or metabolites. And what I want you to appreciate, while this isn't exhaustive, the list gets very long when we think about metabolites. And it's actually not small when you think about proteins as well. And so to actually understand what's going on in a biological system, you have to be able to quantitate or visualize or measure these different components of the central dogma of biology. But there's so many different technologies you can use, and there's actually not much overlap in what they're able to detect. So for instance, the metabolites you're going to be able to detect with GCMS does not overlap very well with, say, CE, LIF, and so on and so forth. So this gets to the crux of my research and really what kind of problems I'm trying to solve, is that no single approach will let you measure everything about a biological system. But yet we need to really understand the molecular basis to understand human health and disease. So I've spent a good majority of my academic career developing multimodal approaches that allow us to combine multiple of these different technologies together to really increase the chemical space that we're able to measure. So as a graduate student at University of Illinois, Jonathan Sweetler, we developed methodologies for performing single cell MS, allowing us to probe a couple hundred different metabolites or lipids within an individual cell in a variety of different mammalian systems. About the second or third year of my PhD, I really was looking at all this rich molecular data and realizing it was very difficult to actually understand it without additional context. And so the first thing we did is actually coupled our rich single cell information with immunocytochemistry. So we can say, oh, I know that this cell is an astrocyte, and then this is its molecular profile. And through this kind of thought process, I've worked with many other talented individuals to couple single cell homology with Raman or transcriptomics or capillary electrophoresis and IR or other MS approaches to really expand the amount of information we can get from a single cell. Well, I really enjoyed this research. I thought it was very important, and it actually tells us a lot about systems looking at them at a single cell level. But as we know, in biological tissues particularly, cells aren't isolated. They're talking to one another. And so it's important to actually look at not only a cell with extended chemical coverage, but actually what's around it, what cells are it's talking to, and what chemistry do those cells possess, and how does its partnering cells or neighbor cells contribute to its chemical diversity. So as a postdoc, I really wanted to develop tissue imaging methodology. So I did a postdoc with Richard Caprioli, who invented imaging mass spectrometry. And I've persisted in this idea of multimodal imaging, where we can image things such as lipids and then combine it with highly multiplex immunofluorescence, spatial transcriptomics, cyclic IF or cell dive, FISH or histological staining. So again, we can put this rich chemical information into biological context in ways that physicians are used to looking at the data, or there's this rich literature surrounding immunofluorescence. And so primarily what I did as a postdoc, and what I'm going to extend into my into the Neumann lab and as an independent academic, is to build these cellular and molecular atlases of different organ systems, both as a function of disease or health. And so we build molecular atlases or cellular atlases. And so what this means is we're similar to a world atlas. So say we have Norway. If we're looking at an atlas of Norway, we know where Norway is in conjunction of the entire globe. We have a very wide view of where Norway is at in context of all the other places in the entire globe. But an atlas doesn't stop there. It doesn't go, Norway's here, this is what's around it, okay. An atlas will actually give you multi-dimensional views of Norway. It will tell you the road systems, the topography, the different rivers and oceans within Norway, as well as the towns or cities that construct Norway. And so this is exciting because it gives us this multi-dimensional, multi-scale view of a system. And so what we're trying to do is perform this for different organ systems, and I'll show data mostly for the kidney today. Although as an independent researcher, I'm trying to really establish myself within the neuroscience community. Although all of the different technologies I'm going to talk about today really aren't just limited to the kidney or the brain. I've published on batteries before. I'm working with materials chemists right now. This is a very flexible set of technologies. And so for those that aren't renal pathologists in the room, the kidney is important because it allows you to filter different contaminants in your blood. Renal failure or the lack of kidney function is really detrimental and you get very ill. And so the kidney broadly can be broken down into 2 or 3 different regions: the cortex, the medulla, and then the renal pelvis, which isn't shown in this cartoon. This right here is a renal pyramid. And so within these different regions within the kidney, there's the nephron, which is the functional unit within the kidney where all of the different filtration occurs. And so this nephron is right here. And there's a glomerulus at the end where your blood is actually filtered in this glomerulus. And then different key nutrients are reabsorbed as the urine or water gets filtered through these different tubules before it's excreted as urine. So it's a very critical and highly complex organ. And so how do we actually think about the kidney in this concept of a cellular atlas? And so we use co-detection by indexing or codex multiplex immunofluorescence. And so this is a fairly new technology where we have an antibody. Now these antibodies are similar to those produced by your immune system, such that you produce these antibodies and they bind very, very tightly to an epitope that they recognize. So in your body, you produce antibodies against different bacteria or different viruses, things that make you sick or foreign entities. Here we hijack this kind of, we hijack this system, produce antibodies that bind to proteins that we're interested in studying. And unlike traditional immunofluorescence, we conjugate an oligonucleotide sequence onto the antibody. And what this allows us to do is add a complementary antibody or oligonucleotide sequence to a fluorophore, allowing us to visualize with light the position of this antibody. So wherever it's bound, it allows us to visualize it. And so by adding different oligonucleotide barcodes, we can actually visualize the location of 2 different proteins. And because we're using oligonucleotide barcodes, just like your cells do every day, you can add and remove very easily these different DNA barcodes, allowing you to cycle different antibody or the different fluorophores, allowing you to visualize different antibodies by even though you're adding all primary antibodies at once. So very quickly, you can understand that by cycling 3 different fluorophores, this will allow us to visualize dozens and even like 100 different proteins within an individual tissue. And for large areas, which I'll show you in a bit, we traditionally work with 10 by 10 millimeter size tissues. So we're getting a large number of cells for a plexity of about 20 to 60 different protein markers. And this is at 500 nanometer spatial resolution because we're working with fluorescence, which is light diffraction mediated. And what's important is why you can use this for a variety of different things. We use this to provide cell type or cell state information. And so to give you an idea of what this workflow actually looks like is we obtain a surgical resection. We trim it to a region of interest or a size that's appropriate. This isn't necessarily what you have to do, but this is how we do it so we have a manageable size of the tissue. We then slowly freeze and embed the tissue to preserve its morphology. So we do fresh frozen, there's no fixation. We section to 10 microns, perform autofluorescence microscopy for image registration, and I'll discuss that more when we start integrating codecs with MALDI IMS. And then we perform codecs and fluorescence microscopy. And so additional kind of workflows that are important to this is conjugation of the barcode to the primary antibody, and then tissue preparation. So the reason these two side steps I mentioned here are important is that you can conjugate a barcode to any antibody. I've worked with both custom and commercial antibodies. They work great. And so you can actually raise an antibody for a protein of interest and conjugate it to a sequence of your choosing. And this is a lot of flexibility and a lot of power. Additionally, we add all primaries to an antibody and then fix it. And this allows us to actually cycle these solvents, these different barcodes, many, many times without degrading the tissue. And I'll show you some data that shows that we actually aren't decaying the tissue over time like you would in a cyclic IF experiment. So this is just an overview image of a Codex IF image from a 66-year-old male. So this is the kidney. And just to orient you here, these little spheres are the glomeruli. Remember, that's the capsule or the end point of our nephron, which is the workhorse of the kidney, where all the filtering takes place. And I've labeled some different protein markers we have labeled here in their relative colors. But what I want you to really take away from this slide is even just visualizing 3, 4, 5, 6, 7 different markers. Look at the diversity of cell types within the kidney, which many people believe to be homogeneous. This is a cross-section of the cortex, and so there's a little bit of medulla here, but largely we're visualizing the cortex. And this is a zoomed out image of our kidney, but we have a zoom in image as well, because remember, we're still imaging at 500 nanometers to 1 micron, depending on the objective. I think this is closer, probably about 1 micron. Allowing us to get a very high resolution image of, say, glomeruli, proximal tubules, podocytes, mast cells, epithelium, for very, very large regions of cells. So we're actually able within this single experiment to visualize hundreds of thousands of cells within this patient. And while we get very pretty images, and I'm showing you a composite, again, we're looking at a bunch of different protein markers, not just the 6 or 7 I showed, allowing us to probe things such as CD7, which tells us if a tissue is diseased or if there's any immune cell infiltrate, as well as like functional cells like uromodulin or aquaporin 2. And what I really want to hit home with these series of thumbnails is that a traditional immunofluorescence experiment maybe gives you 4 to 7 protein markers. But each one of these thumbnails has a different distribution within the tissue. And so you're really losing a lot of the structural information within a tissue or cell type information if you limit yourself to just 4 to 7 markers. And really, I think spatial biology needs to start moving towards these higher plexity experiments to really get a more full understanding of the tissue microenvironment. And so to just give you an idea of why, how we can use this information broadly, and then I'll go to a second example, is we can look at 9 markers that stain glomeruli. These are the same 4 glomeruli, just 3 images by 3 and 3 different colors. And so just comparing the relative difference between red, green, and yellow pixels, you have diversity within the glomerulus and what proteins it's expressing and where. And so, for instance, this glomeruli is not really expressing nested at all, while the adjacent 3 are. And so some would argue that maybe this is a difference within the depth of the glomerulus that you're probing, although more or less these are the same size in circumference, so they probably are actually similar in depth. And so what this gets us to is that we can use these highly multiplex immunofluorescence approaches to look at heterogeneity within structures, not just individual cells. And while we produce many pretty pictures, we can actually use this information to start accessing cellular neighborhoods and building cellular neighborhood analyses. And so these are Spearman correlation coefficients, and this is just a heat map. And so you can look at the numbers or completely ignore them. because I've recolored the values. But this is a semi-quantitative way of actually assessing different neighborhoods throughout our entire tissue section for, again, hundreds of thousands of cells. So to orient you, cells within this heat map that are correlated with one another are red, and the more red two markers are, the more correlated. Uncorrelated are in white or gray, and anti-correlated are in blue. And so I've taken our large panel of 20 and actually reduced it to several markers because it's actually painful to look at a 25 by 25 matrix. This is only 9, and that's still like a lot of data to look at. So how we actually calculate these is we have a cell type in the middle that is expressing nestin. We then calculate a radius 20 microns out from this cell, which is about a 2-cell layer, and ask ourselves what cells are within that 20 micron radius from this nestin cell, and then build the correlation coefficient to allow us to understand how often different cell types are within each other's neighborhoods. So to give you an idea of what this means practically is nestin is in a neighborhood with cells expressing CD31 48% of the time. So this is actually pretty neat. And so we can do this for glomeruli localizing antibodies and tubular localizing antibodies. That's partly why there's not many anti-correlated things except for smooth muscle actin, which tends to build up in glomeruli that are diseased. But what we can do is actually start comparing our normal kidney information and cellular neighborhoods with like disease states such as diabetes and diabetic nephropathy. And so diabetes is critical, it's important. Most people know someone with diabetes or know someone who knows someone with diabetes. And in 2015, they figured out 1 in 3 Americans will have diabetes, which is a staggering number. And so I would usually think about diabetes in the context of the pancreas. Diabetes is the #1 cause of kidney failure. And part of this is the increased sugar content causes your glomeruli to secrete proteins that are critical. And that's why one of the diagnostics of diabetes is measuring the protein in urine. And so what we did is we actually performed codex immunofluorescence on healthy and diseased human kidneys for multiple patients. I'm only going to show examples of one, although we have data from multiple patients, and I'll talk about the composite data in a little bit. But this is an example diabetic nephropathy sample from a 65-year-old man. So this is a similar age group and similar demographics to the individual that we looked at earlier. And we're also within the cortex of this kidney. But what I want you to appreciate is it's really hard to actually pick out the glomeruli. They actually are shrunken in nature and they just look diseased. They're often more sclerosed and so on and so forth. And so just visually, you can see how different diabetic or diabetes really affects the kidneys compared to a healthy counterpart. And so we can compare the neighborhoods within healthy diseases and diabetic nephropathy with our Spearman correlation coefficients, and you can compare the numbers. But what's easier with this is to actually survey changes in color. So for instance, this cell right here goes from white to blue, and again from white to blue, saying that there's now an anti-correlation between smooth muscle actin and nestin and CD34 that we experience in diabetic nephropathy as opposed to healthy individuals. And so this is a composite of 5 healthy and 5 diabetic nephropathy samples. And we have a survey about 100,000 to 200,000 cells per sample. And so this is a semi-quantitative way to actually measure broad changes within the cellular microenvironment without having to actually look at individual cell neighborhoods. And so by looking in what correlation coefficients change drastically, this lends us to the understanding of perhaps what other cell types are actually differentially affected by diabetes. So for instance, things become negatively correlated. Many things that were moderately correlated are now non-correlated, and so on and so forth. You can then do that for glomerulus and then tubular localizing tissues, just to give you an idea that we're not limited to one thing. We can look at many different markers and many different correlation coefficients. And so what I've hoped I've shown you is that you can perform codex IF on fresh frozen. I didn't actually show the FFP data here, but you can do it on FFPE or fresh frozen kidney. I've done it on other organs as well. You're not really limited on the organ front. And I showed 9 codex cycles, which ends up being about 27 antibodies. We've done up to 60, so it really allows you to visualize a high dimensionality of your tissue. So this is cool, and I think cell type and cell state are really important to probe, but we're actually missing a lot of the metabolomic data. And you can't actually get at that metabolomic data with immunofluorescence. So we'll briefly talk about matrix-assisted laser desorption ionization mass spectrometry to study small molecules. And so small metabolites are fundamental to different biological systems. Here's our central dogma of biology that I referenced earlier, where DNA goes to RNA, RNA goes to proteins, and then small molecules actually interface at each of these different steps. And to give you an idea of what I think about when I say small molecule or small metabolite, so we're talking about like small sugars like glucose, fatty acids, precursors, amino acids, or like cholesterol, things of that nature. Cholesterol is hard to see with MALDI, but we'll talk about that later. And so my slide loads. Matrix-assisted laser desorption, ionization, imaging mass spectrometry, and MALDI IMS, I will never say that full name again in this whole talk because that's just a lot of words, is actually similar in sample prep to our immunofluorescence experiment at the beginning, allowing us to just acquire a tissue resection from a patient, section it, or embed it, freeze it, and then section it. But at this point, we apply a matrix, which is a small organic molecule. What you can kind of think about this matrix, or this MALDI matrix that we're applying, is it kind of functions like an explosive. And so we spray it on wet. You can sublime it. There's a lot of different ways to do it, but for sake of discussion, we spray it. And this allows the matrix to extract different molecules that we can detect with our mass spectrometer. It then co-crystallizes so that you aren't just putting a liquid into a vacuum, because you would then pull a bunch of stuff into your system and get really dirty. So this dries, it co-crystallizes with the metabolites it's extracted, and it's actually very stable. The matrix we apply is an antioxidant, and so it actually preserves a lot of the chemical and molecular construct of our tissue. We then perform MALDI MS analysis. And so for those unfamiliar, we can actually build an image by shining laser on a pixel or a point on our tissue. And then we get ionization of the matrix with the co-crystallized metabolites. So this is where this kind of serves as an explosive, where you shine a laser and the matrix absorbs this laser line and then makes a giant explosion where you get a plume of different ions, both of your matrix and both of your tissue and molecular information. And by rastering this laser across our tissue, we collect spectra at each discrete location, allowing us to build ion images of many different molecules within our sample. And I'll briefly talk about mass spectra. I think I actually have it in the next, I don't have it in the next slide. For those unfamiliar looking at a mass spectra, each one of these peaks is a discrete molecule that you actually are detecting within your system. And by actually using different software, you can actually visualize the relative abundance of those molecules throughout your entire tissue, giving you an image of actually the location of each of these detected molecules. So in this paper, we also do some ion mobility separation with traveling ion mobility spectrometry. And so unlike other types of ion mobility, you don't actually use a drift gas to separate your different ions. You use an electric field. So you use a drift gas to actually push your ions through this TIMS funnel and use an electric field to actually track them. And then by lowering this electric field, different ions will elute based on their apparent shape, allowing you to actually separate in the gas phase isomers or isobars. And this becomes super critical in metabolomics, where there's many different isomers, or it's in when identification is actually non-trivial. And so here is an example of the small molecular mass spectra that we can get off our TIMS system. And so I actually, in my lab, have a timsTOF Flex. This was not acquired on the one that was installed because we clearly published this 2 years ago. But we can get the similar data on the one downstairs that's currently operational. And so this is average mass spectra for a large section of a human kidney. And we have the difference without and with eye mobility separation. And so you actually get higher, you lose some of this small molecular information with eye mobility, which we're still trying to figure out. And we have a larger abundance of these higher mass species. And so there's benefit to actually do both with and without TIM separation, because I get asked a lot why we just don't acquire eye mobility for every data set. And it's because you get slightly different things. And part of that is like the pressure within the TIM cell and so on and so forth. And so just to give you an idea of what kind of molecules we're able to detect, we run the gambit from a small m/z of 86, which is a choline minus water, to a fragment of heme at 616. So this is our molecular sweet spot. And we again see some vitamins, some lipid precursors, tripeptides, so on and so forth. And again, we're acquiring this data within an imaging context. And so here on the left is an autofluorescence image of the kidney, where you can make out the cortex, the medulla, and the renal pelvis right here. This is something to help orient you as we go through different images of kidney. So remember, in our cortex, there's some glomeruli, which you can kind of make out here, these little circles. We have this co-register with microscopy, but you'll have to believe me that these are glomeruli. And so what this technology allows us to do is, say, measure choline at an m/z of 104, we've identified through MSMS, and actually see that as a higher relative abundance, as depicted by yellow, within the medulla and renal pelvis, as opposed to the cortex, which has some amount of choline in it, but it's at a lower abundance as depicted in like the blue. Black is actually our 0 here, and yellow is our 100%. So to give you an idea, we go from low to high abundance. You can also see things like acetylcarnitine, which has a high abundance in the medulla. We have a couple unknowns, and then drugs like this one at 280 that are within the renal pelvis and medulla, and so on and so forth. And so while MALDI primarily has not been used for small metabolite analysis because of the interference with the matrix you're adding. So I mentioned earlier that this is a small organic matrix. And so it's often isobaric with molecules that you're interested in. And so if we don't have ion mobility and we just have a composite, we get this ion image at this m/z that I can't remember. I think it's about 150. But with ion mobility, we get a separation between this species I'll call alpha and the species I'll call beta, where we have a high abundance of this alpha because it's in more pixels, and a low abundance of this this interference or this matrix background that's localized here. So using ion mobility, we can actually recreate these ion images with just these species alpha and beta. And so this allows us to look at GPC with high resolution within the cortex and medulla with high specificity without this contaminant, allowing us to get more true localization within our image. And again, we can visualize the matrix right here. And so while exciting and cool and really helps make MALDI applicable for small metabolites, I discussed earlier briefly that the metabolites or small metabolite molecular space is very complex. And I'm sure Oliver can attest to this, and individuals in Oliver's lab as experts in metabolomics. And so using ion mobility, we can take this composite image and it looks like, okay, great, we have a metabolite at, I want to say this is 210. So we look at just m/z 210 and we get this composite image where there's some hot spots and it more or less has an equal distribution through cortex, medulla, and renal pelvis. But again, using ion mobility, there's actually 4 different conformers within the ion mobility space, allowing us to extract their ion images, allowing us to have a more true understanding of where these features are actually localized. So to give you an idea of how actually different these ion images are, because alpha and beta are actually inverse of one another. So alpha is primarily localized in the renal pelvis and beta is in the cortex largely, not even really in the medulla. Whereas gamma and epsilon are more or less throughout the entire kidney. And so all of this chemical information is lost in the composite image, which shows why there's advantages to using ion mobility to study the metabolome. And while I'm showing small metabolites here, the same can be said for peptides, lipids, and proteins. Ion mobility can allow you to help separate out these different complex features. What I want to point out though at this point is I have alpha, beta, gamma, and epsilon. I have no idea what these different features are. Metabolites are difficult to ID in general, and the more complex these systems are, the harder it is to ID. And so I actually have not gone back and actually been able to identify these, although I have an idea of how I would go back and do it if it turns out these are important for atlas studies, which I'll talk about a little bit in the integrated data. But using standards and things and ion mobility drift times or CCS values, you can actually start putting together what these different conformers may actually be. So I hope I've showed you that mass spectrometry imaging allows you to actually visualize the location of many different features. Again, we're looking at hundreds to even thousands of different features within our biological systems. And by using ion mobility, other mass spectrometry-based separation tools, we can get a better understanding of the information within our data. And so with that, I've talked about and shown why codex multiplex immunofluorescence is useful and what kind of information we can get from that technology. And I've discussed multi-IMS and what it can do. But I'm a multimodal mass spectrometrist, and we actually integrate these two data sets together to really expand what we're able to look at. And so I'll show two different examples briefly of how we use this integrated data to better understand biological systems. And we'll start with HubMap. And so this is actually our Kidney Atlas program. HubMap is an NIH-funded program. I'm the Human Biomolecular Atlas program. I'm actually no longer part of it, but it's been really fun, and I've learned a lot and still keep up with a lot of the different collaborators I've made there. And so our goal with this is to, again, build a cellular molecular atlas of the human kidney at different spatial dimensions. And so we can take an unused donor kidney, section it into different, or physically cut it into different regions. We then have several that are fixed and paraffin embedded. So this would be our gold standard histological point of reference. And then fresh frozen for our various mass spectrometry analyses. So by performing serial sections of dozens or even hundreds, and using autofluorescence, we can register IMS of small molecules and lipids to histological stains to MXIF, and do a battery of different imaging modalities that are inherently registered with one another. We can also use this to build 3D maps of our chemical information, which I won't discuss today, but I'm happy to discuss offline if anyone's interested in 3D imaging. So to give you an idea what this multimodal data set looks like, and these are actually publicly available in the HubMap data repository, and I can link to that later if anyone's interested to actually look at all this data. We have autofluorescence, and we collect this before imaging mass spectrometry, and this serves as a way to do segmentation of different molecular features, as well as a port for registration of our different modalities. We then have lipid analysis or small metabolite, depending on what it's labeled, for a variety of different lipids. So here's an example going from black to yellow or low abundance to high abundance of a kidney for this sphingomyelin, where we have high abundance within the glomeruli in different regions within the cortex and some hot spots within the medulla. And again, I'm showing 3 different lipids here, but this is of like about 1,000 ions. We can traditionally identify about 200 per sample. We then can perform histological staining or periodic acid shift-based staining after multi-IMS. And as you can see, there's not really the effective laser. You don't see the ablation spots. And so you can get a nice histological image. And then on a serial section, and you can even do it on the same section, I'll not talk about that today, but that's of active research in my current position, is performing multiplex immunofluorescence. So we perform autofluorescence first and then multiplex immunofluorescence. And this autofluorescence on both of these sections allow us to actually register the tissues together. And so to give you an idea of what this actually looks like, and the different timelines required for this kind of multimodal approach, I have the schematic, and it's part of the STAR protocols where there's detailed protocols for performing this. But we'll look at the data, but it's actually important to know that it takes an amount of time to actually produce data, particularly for each patient. So we resect the kidney, we divide and preserve it. The preserving takes about 30 minutes. We have an FFPE and a fresh frozen sample. We then perform histological assessment with Agnes Fogo's group at Vanderbilt Medical Center. She's an expert renal pathologist. And this takes about 1 or 2 days because they're manually going through each kidney by hand to actually locate regions of disease or disorder. And I'll talk about that in a second and why that's actually critical. So this is non-trivial. It takes a day or two per patient because we're, again, looking at large samples, and you have to have and look at very high resolution to sometimes see these different diseases. And then we have fresh frozen, where we perform autofluorescence in about 30 minutes, imaging mass spectrometry for a day or two. You can make this go faster if you look at smaller areas, but there's not a whole lot of fun in that. And then Codex multiplex immunofluorescence takes 1 to 2 weeks. And so think about, for every patient, there's at least like a 2-week period of time where this is getting analyzed. And while IMS and Codex can be done serial or like in conjunction because these are different tissue sections, there's projects where I've done both of these on the same section, and that takes even longer because you're having to do this in this and you can't run them in parallel. And so our atlases that are publicly available are composed of, I think this is 23 patients. We have 45 now, I think, on the thing, but I'm talking about the subset. And so we have a sample ID where we've ID'd them, and we have their relative age, their sex, their race, and their BMI. And this tells us a lot about each of these samples. As you can notice in this table, I don't want you to have to read all of it, but we have pretty good coverage of men and women. We lean very heavily on white patients, although that's something I would like to fix in my my current position, is to really expand our race coverage. And then we have ages ranging from 20 to 77, although most of our patients are within their 50s. And we run the gambit for BMI, so we have normal BMIs all the way to morbidly obese. And so this is all important because each of these different characteristics of our patient tells us something about them. And allows us to actually discriminate the differences in sex and BMI, which I'll discuss later. And so in collaboration with Agnes Fogo at WCMC, we can take images of these different tissue sections and assess their percent cortex, percent medulla or other tissue, as well as assess them for different diseases. And so this serves 2 purposes. One, these kidneys have been assessed to be relatively healthy, but the kidney gets more disordered or diseased as you age. So what's healthy for a 77-year-old is not healthy for a 20-year-old. So that's important to note. Additionally, just because you have a healthy kidney doesn't mean you have a sclerosed glomerulus somewhere. And so it's important to know where these areas of different disease and disorder are so we can actually accurately represent them in our atlas. And so we're actually probing several different diseases such as arteriosclerosis, glomerulosclerosis, different lesions, so on and so forth. And so we're taking this for over 20 patients, and we have even more that the data is actually based on now. But I'm going to use a single patient to show you what a single data set would look like, and then some composite data at the end. So this is Codex data from a 44-year-old man. And this is the division between the medulla and cortex, where the medulla is on this side and the cortex is on this side. And so going back to our nephron, our functional tissue unit, these are glomerulus, which is the capsule of the nephron, and these are different tubular segments. I have each of the proteins we're staining down here, but they're really not important at this point. What's important is the number of colors you can visualize, where each different color is more or less a different cell type. And so within the codex of even a portion of our panel, we're getting a lot of different cellular information at, again, high spatial resolution. And so we can use this, again, to build cellular neighborhoods for all of our patients. So this is actually a composite of our 20-something patients, allowing us to look at Spearman correlation coefficients in bulk tissue for, I think this ended up being about 3 million cells. And so what's interesting is in so many cells, there's correlation coefficients that are quite high. So say PARP1, cells expressing PARP1 are 77% of cells expressing PARP1 have a cell expressing CD93 in its cell or in its neighborhood. And so quickly you can see that there's some cell types that are often found in each other's neighborhoods, despite profiling 20-something patients and millions of cells. And while this is cool and gives you a really broad semi-quantitative understanding of the cellular neighborhoods for glomerular markers as a composite of all of our patients, we can separate this based on demographic or characteristic of our patients. So we have male versus female or high BMI versus low BMI. And just very quickly, you can see that there's some KDR is often anti-correlated with many of these glomerular markers in men, but is modestly correlated or uncorrelated within women. And moreover, most of these correlation coefficients are largely within margin of error. And so while there's some differences between men and women, they're not nearly as drastic as those between individuals with high BMI and low BMI. So very quickly, you can appreciate that by taking this information from millions of cells, we have an idea of what different cell types and cell states are actually affected, say, by high BMI, such as cell types expressing vimentin, and how this maybe affects cellular reorganization within our tissue. And so we can then, again, couple this with the rich molecular information with multi-mass spectrometry. And so here's some lipid data, although, again, we're not limited to just lipids. So this is an average mass spectra of a human kidney, where each of these different peaks is a discrete molecule we're able to detect. So you can see each peak, there's probably hundreds of peaks. You can zoom in and even see more peaks. I've gone through and labeled some of these. This is in positive mode. We collect both positive and negative modes. So we have coverage of many different classes of lipids, such as sphingomyelin, PAs, PCs, PEs. In negative mode, we have even more. And so this is to just give you an idea of what quality of spectra we're able to obtain from these kidneys for, again, all 20-something patients. So to orient you, this is a serial section of the kidney tissue that I showed a Codex image of earlier. It's just rotated 90 degrees because I like making my slides more complicated than they need to be. So this is a division between the medulla and cortex, where this is our medulla and this is our cortex. Mass spectrometry, again, isn't quite as high resolution as immunofluorescence. We're at 10 microns right here, which is why it looks fuzzier than the Codex image. But it's the same region and it's very large. And so I'm, in this image, visualizing a lipid recolored in red, green, and blue, where black is 0% abundance and bright red would be a high abundance of this, this PA lipid. And so as you can appreciate, there's very little pixels that are colored just red. There's maybe some orange here. And most of it is a mixture of purple or pink or yellow, which is not any of these lipids entirely. And so what I want you to appreciate is just looking at three lipids, the distribution of them is quite remarkable in the kidney because you get this mixture of colors. So the relative abundance and mixture of these three lipids allows you to visualize the glomerulus, different tubules, different proximal tubules, so on and so forth. And again, we're looking at hundreds of features here. And so these are just 9 to give you an idea why each of these different lipids are important to be able to identify and visualize. And just so you know, I'm not lying, here's a mosaic of what I don't even know, 6 by 11, so 66 ion images of different features that we've identified as lipids. I didn't put the names on them, and these are only, again, a small subset. But each one of these different thumbnails are different and have different localizations throughout the kidney, where on the right is the cortex and on the left is the medulla. And so by doing a bulk analysis, you'd actually miss this spatial context and diversity of where these lipids are located. And so again, I'm talking about integrating these two rich data sets together. And so this is where we start actually getting into cool data integration stuff. I'm kind of a nerd. And so we have our cartoon of a nephron, where again, we have our glomerulus with different tubules like the proximal tubule, loop of Henle, distal tubule, and collecting duct. And using a combination of multiplex immunofluorescence, which allows us to label these different features and cells within these features, as well as our mass spectrometry, we can build molecular profiles for these different functional units. So using machine learning, we can find the features that best define glomeruli which features on the left, or these are actually all features that using machine learning discriminate glomeruli compared to all other features. And we use violin plots to actually indicate and to visualize the distribution of this molecule within our 20-something data sets and millions of pixels. And so, as you can see, the sphingomyelin has a normal distribution within glomeruli that's higher than all other pixels, and so on and so forth. And we can do this for proximal tubules, collecting ducts, distal tubules, anything that we have a fluorescence signal for, or a combination of antibodies that we can use to visualize them. And so where I get excited about this is we're not limited to looking at the entire kidney. We can start specifying or comparing different demographics. So here we're looking at differences between male and female, or kidneys for male and female patients, where the left is male and blue and the right is female and pink. And we can use, again, machine learning to see which features differentiate men versus women. So here on the left, we have this, this PE, which has a low abundance and then a mid to high abundance within men. So it's a bimodal distribution, where it's only a single distribution within female patients. And so, so on and so forth, we can actually look at these different markers and see, oh, these are the largest differences between men and women. Can take this a step further and actually look at molecular neighborhoods. So this is similar to our cellular neighborhood, where instead of a cell at the center, we have a pixel. And we look at the neighboring pixels around it to see if there's lipids that are highly correlated or negatively correlated with one another. What's interesting is we have lipids that aren't actually related to one another, such as this PA and this PE, that are highly correlated. So the radius of this circle indicates its Spearman correlation coefficient. We have the correlation coefficients kept, they're just, it makes it hard to actually visualize. And so this is a small subset of the lipids we're able to probe, but we're seeing some really interesting differences between men and women, where we have a moderate correlation coefficient and then an almost non-existent correlation in women. So we can take this a step further and start looking at obese versus healthy individuals. This is actually where the data starts getting interesting. So again, using machine learning, we can build these profiles and we can build these violin plots for all of our different lipids that we've detected. But this first lipid is interesting. It's an oxidized phosphatidylcholine. As you can notice, it's got a relatively high abundance in obese individuals and a relatively low abundance in healthy individuals. And it's bimodal in obese and probably got a long distribution within healthy. And so what's interesting about this lipid is it actually didn't show up in a database initially. And so I was very confused what this was, but it was the #1 discriminator of obese versus healthy patients. And so this oxidized phosphatidylcholine has been noted in literature to actually accumulate in kidneys suffering that are in the middle of renal failure. And so it's interesting to note that it is higher abundance in obese individuals. And by MSMS and validation with a standard, we've actually been able to identify this as this oxidative phosphatidylcholine. And it is in fact in a higher abundance in obese individuals, indicating that perhaps these kidneys are more diseased than their healthy counterparts. What's interesting, though, is there's some pixels within healthy individuals that have a high abundance of this oxidized phosphatidylcholine. And so what we're doing with our renal pathologists is actually finding these pixel locations where there is a high abundance and seeing if those are areas that correlate with high disease. And this is a little preliminary, but it looks like that they actually do. And so this is interesting to see that, one, this marker is shown to be highly correlated with renal failure, but also you see it in healthy and obese tissues. Moreover, if we look at this PE lipid, we have a very tight distribution within male or within obese individuals and a fairly tight distribution within healthy. And so this is actually cool that lipids actually vary this much within obese and healthy individuals. And just to briefly, I meant to mention this earlier, these lipid species are validated with MSMS and orthogonal liquid chromatography experiments. If we don't have teleinformations because we didn't really get specific tail information, or it was a mixture of a bunch of different tails. And so we're just, we're being careful here. And again, we can calculate neighborhoods and look at differences between obese and non-obese patients. So finally, for this project, I've talked about bulk kidney, but we can actually go back to our functional tissue units, such as glomeruli, proximal tubules, so on and so forth, between obese and healthy. And we can do this as a function of sex or whatever, and start looking at how these cellular neighborhoods change, as well as their molecular neighborhoods and molecular distributions. And so this oxidized phosphatidylcholine is actually still the #1 marker of obesity when you're looking at distal tubules. So not only do we know that this oxidized phosphatidylcholine is critical or a high discriminator for obese versus healthy, it's a discriminator for different functional units, such as the distal tubules, allowing us to get a better cellular context for this rich molecular information. And and this is cool, so I hope I've showed that we can do this, and that while there's a lot of data here and we're still actually trying to understand it, that we get a really complex data set that spans from single cells all the way to bulk tissue. And so finally, I want to briefly talk about the host immune response. I think I'm going to start speeding up just to give you an idea of the flexibility, again, of this multimodal approach. And so staph is important. It costs billions of dollars a year. It kills a bunch of people. It's very important. Staph is interesting. Here's an electron micrograph of staph, and what I want you to appreciate, there's no flagella or cilia, so is completely stationary. And because it's stationary, it forms these abscess structures, and there's immune infiltrate, and it just looks very diseased. And so this is an example, histological stain of an abscess within a kidney of a mouse. And so when we think about the immune response, the host responds to bacterial infection. We have innate immunity, which is made of these different cell types, which is very rapid and not very specific. And then we have adaptive immunity that's slow to respond, but more specific, such as these antibodies that keep coming up over and over. And so if we think about the host path interface, it's a giant disaster. You have bacteria producing things and producing things because other bacteria are there. The host is also fighting by producing a bunch of these different cells, and everything's producing chemicals, and it's quite a disaster. But it's super important to understand this interface, because some people recover from these bacterial infections with or without antibiotics, and others don't. And so this gets to, like, how do you actually understand the host pathogen interface? And so I think by using these highly multiplex immunofluorescence and MALDI imaging approaches, we can start understanding and unraveling this complex interface. And so we have mice infected with Staph aureus. We section to 10 microns and do our procedure as previously described. And so this is what our MALDI IMS data looks like. Again, we're looking at 3 lipids here in RGB, or our red, blue, green, where we have a marker that really lights up staph, this plasmalogen. It really lights up this abscess. We have the medulla here. This is a mouse kidney, so it's much smaller, and then the cortex here, and then the inner medulla. We're not limited at looking at 3 lipids. We have a bunch, and so these are just different distributions of different lipids within these kidneys. And what's interesting here is that there seems to be some abscess heterogeneity within the local, or within the possession of this plasmalogen here, which is cool. And so again, because we're untargeted and we can detect hundreds of thousands of features, we can detect lipids that are localized primarily to the mouse or the native host. Lipids that are shared between immune cells and abscess. This should be abscess lipids, not staph. There's some staph lipids, but I'm not going to go into that here. And there's lots of lipids that are shared between the abscess region and the native healthy host microenvironment. And so this is required without ion mobility. And so future steps include using ion mobility to separate out the lipids that are probably more localized to the abscess as opposed to everything else. And again, we have this inherently registered with our multiplex immunofluorescence. And so this is a larger panel. It's actually 60 at this point. And this is 6 of our 60 markers, allowing us to, again, visualize the different abscess regions, the medulla, and the cortex. And so we zoom in. I apologize, the color switches. I forgot to change that out. This is a better, long story short, I redid the multiplex immunofluorescence, and it looks better and it's higher signal to noise here. But this allows us to actually look at this microenvironment within the staph or within the abscess. The staph is colored in red, so we can see the staph colonies here. This is the relative abscess region where we have, these are monocytes in pink, as well as calprotectin in blue. Calprotectin is expressed during the disease process, as well as a layer of B cells. And what's interesting is you can have these abscess features where most have a staph nidus in the center, but there's some that don't. And this is, I think these are T cells, if I'm remembering correctly. And so from going from different regions within our kidney, we have different stages of abscess formation, as well as a different immune response. And moreover, we can look at a tissue such as in blue, which is farthest away from all of our abscess, but we even see monocytes starting to infiltrate this healthy functional tissue that we have labeled. And so again, this is serial to our MALDI-IMS, so we can start looking at molecular features that actually correlate with these different abscess or cell types or immune cell stages. And so using machine learning, we can actually build profiles for, say, progenitor cells, neutrophils, or B cells, using a combination of multivariate statistical approaches and different machine learning technologies. And so I'm only showing an example of 6 here, but as you can appreciate, we can start looking and finding which features best define these highly complex immune states within our tissue. And so hopefully I've shown that MALDI-IMS and Codex can be used not just for human kidney and atlas building, it can be used in the context of disease and disorder. And while both of these were kidney, we've looked at things like spleen and heart, and I've looked at brain before. So we're a very flexible suite of technologies. And so with that, I'd like to acknowledge all of the people that have helped with these various projects. Good science isn't done alone, and so the individuals italicized and in bold have really contributed a lot to this research, particularly Heath Patterson and Jamie Allen. Heath has done a lot on the computational end, and Jamie has done a lot with on the sample prep end, as well as all of our collaborators and our funding mechanisms. And so in the last couple minutes, I just want to share what I'm actually doing here at Davis. So I'm switching gears a little bit because I really enjoy neuroscience, and I would like to study autism. And so 15% of autistic cases are because of known genetic disorders. And so the cynic will say, well, clearly we just don't know about the other 85% of genes responsible for autism. But I think it's more complicated than that. And I think by using this suite of highly multiplexed imaging approaches, we can start unraveling the molecular and the cellular information behind autism. Well, I think that this will work or will provide interesting information is most people think of autism as a spectrum where there's less autistic individuals and more autistic individuals. And this really doesn't do a good job explaining the complexity of this disease. So individuals are each different, and they're better visualized by this radar plot, similar to like a personality plot that you would pull up on like Facebook or something, you know, those quizzes, where there's individuals who are above average with sensory skills and perhaps below average on motor skills, and this varies from individual to individual. Well, if we think about it from a neuroscience perspective, different brain regions actually control your sensory input or how you interpret sensory input and your motor learning and your language learning and so on and so forth. So I think by really understanding how cellular neighborhoods and their molecular features are disruptive in these different areas as a function of whether the individual is above or below average will actually tell us a lot about the learning mechanisms behind autism. And so as a new assistant professor, I have a lab now. It's kind of cool. I'm in the Department of Chemistry. Here's a token view of our building. You should come visit me. My office is in 5465. It's funny because I didn't actually know what being a professor is going to be like because they hand you an empty room. And the first thing I did was make the room more empty by knocking out some cabinets and stuff to make rooms for fridges. The instrument's installed. It's ready to go. Don't Don't look at the red light. It's actually blue right now. It's happy. And we're in the middle of organizing and doing dishes. And I'd like to especially thank Sam and Kyle. I don't have students right now, but they've been fundamental and or they have a lot of pity. They're helping me set up my lab. Kyle especially has done so many dishes, and he's an incoming graduate student. Sam is in someone else's lab and interested in mass spectrometry, so he helped set up the instrument. So with that, thank you for listening to my talk. And if you're inspired or think any of this is cool, feel free to join our team as students or collaborators. We're always looking. We always will be looking. And then here's my email in case anyone's excited about it. And so I think I maybe went over a tiny bit, but I'll take questions if we have time. Thank you for your attention.
Thank you so much, Elizabeth. That was colorful. I think the most colorful talk we've ever had in terms of colors. Yeah, that is very interesting, and especially your notion that people think that kidneys have like more homogeneous. You certainly have dispelled that notion now. Nobody who watched it could now again say kidneys are all the same. As usual, our chat and our question and answer is led by our academic outreach coordinator, Dr. Aparna Vania. Aparna.
Great, thanks, Oliver. Great talk, Elizabeth. Really loved the presentation. Jumping right in, could you please compare your antibody-based protein imaging technique to using MALDI imaging with Ambergen antibodies that have photocleavable tags? It looks like your codex system might be better, more resolution, etc. Could you comment on that?
Yes, I can. So I hesitate to say one is better than the other. So I think a lot of, this is an aside, and then I promise I'll answer the question. I think a lot of analytical chemists get in the mindset that their technology is the best and can be used to solve all problems. And I think that that's really underselling the power of every analytical approach. And so I'm an expert in multi-mass spectrometry, but early on I figured out that immunofluorescence is important. So I don't think any technology is inherently better in one way or the other, but I think they can be used for different things. So Ambergen's cool, and for those not familiar, you use an antibody conjugated to photocleavable mass tag, and it's a peptide, I believe, although you can use a variety of different things like metals. And by using light, not the MALDI laser, but ambient, it's not ambient light, I think it's UV. They just like have a UV lamp. It'll cleave off this mass tag that you can then detect with a mass spectrometer. So the key difference within these two technologies is I use antibodies and fluorescence, which gives me higher sensitivity in a lot of regards, depending on your microscopy system, and better spatial resolution. But where I'm really hit is that you have to cycle, because most modern microscopy systems at best can look at 7 different fluorophores at a time without spectral overlap. So Ambergen, because you're using a mass tag and a mass spectrometer, especially if you're looking at like a timsTOF flex, has high spatial or mass resolution. And so you're limited at that point for the number of mass tags that you can just produce. And so say you can separate things half a Dalton apart, and you measure from, I don't know, 500 to 1,000 m/Z, that's a lot of mass tags you could potentially be looking at. And you don't have to cycle them on and off, so you're not damaging the tissue. And so while codex is considered non-destructive, we have issues going beyond about 40 cycles. Because the tissue starts to disintegrate, whereas you're not going to have that problem with Ambergen. That said, if you have a microscope near you, it's much cheaper to do a cyclic IF experiment, but I think they're complementary. You can also get your mass tags plus lipid information in the same sample. And what's interesting is I talked to the Ambergen people at a conference last week, and I actually was like, this is cool, but what if you added a fluorophore to it? So you could do both, because then you could start seeing how your ionization efficiency is affected by all of your antibodies and things of that with fluorescence microscopy, because fluorescence is more quantitative, and there's no reason you can't actually just add a fluorophore in addition to the mass tag. And so that's actually what I'm going to start working with them on, is to see like how those two technologies compare. And if there's any like ionization effects occurring or so on and so forth. I don't know if that answered the question, but those are my thoughts.
Yeah, I mean, it definitely, kind of explained, why you do things and why you choose certain antibodies and such. I'm just surprised, like, it's just astonishing to me the amount of information you get. Like, you get so much. Why, moving on to the next question, why is there no automatic data dependent MS-MS schema in MALDI-MS?
That's a great question. So the answer is, I know Vanderbilt is working on it. I wasn't part of that project. So the short answer is, I think similar. So if we think about the SIMS community, they didn't think tandem MS was important for a really long time. And part of that was because of the instrumentation limitations. And they just, I've gone to enough SIMS conferences where I'm like, everyone gets excited because they have MS-MS for the first time. And I'm like, it's 2022, guys. But you can say the same thing for MALDI, right? Like, we should be doing much better on our identification and automatic fragmentation and stuff. And we're now starting to get into that. So I know Vanderbilt's working with Bruker. I'm working with Bruker of how you can actually do like a DDA kind of experiment. And there's ways to subdivide your pixels where you can get, say, like a full spectra in one corner of your pixel, move over, and then start doing your fragmentation. And preliminary results are indicative that we can do about 60 precursors, maybe 100. So like, this is, for those who have ever done MS-MS with MALDI, slowly, one shot at a time and just nailing your tissue, this is actually much better. But we're still working on it. It's an area that's important. And we're just recently starting to work on it.
Great. And by SIMS, you mean single ion monitoring, or is that a different
secondary
acronym?
ion mass spectrometry, sorry.
Oh, okay.
With
Okay.
like ion beams, and they still use sectors. Like, it's super powerful. I love SIMS. They just, there's a lot of reasons why it's hard to put a quadrupole at one of your ends of the SIMS instrument because it's really high energy. But because of that, they're like, look, we have 200 nanometer spatial resolution or 50 nanometer spatial resolution. But they don't have any MS-MS and everything's fragmented everywhere. It's very fun to actually think about how you mix these different technologies together to really understand them. And we've actually coupled SIMS with MALDI to get a better understanding of like what molecules you're actually detecting with SIMS.
Oh, that's awesome. Yeah, I mean, not just to explore what they do, but also how they teach us more about the molecules that we're interested in.
Oh, yeah.
Can you discuss approaches for ways to collect data longitudinally, say over months to years, with the goal of combining all that data into one data set? How would that look for a large project, for example?
Oh, yeah. So the HubMap data I showed you was actually acquired over 3 years. So I started at the beginning of my postdoc and finished sample collection actually when I had this position. I didn't quite sign the papers yet, but that was the end of the sample collection. That's at patient 45, or like 45, I think. And so there's lots of things to think about. And there's lots of ways to handle it. You're not going to eliminate all technical variants and all person variants. It's just impossible, especially when you're going from like a surgeon resectioning to sample freezing and embedding. I've gone in at the middle of the night to embed a sample. So like, I can almost promise you at 3 in the morning, my embedding is not as good as 9 in the morning. And like, likewise, we get things flown in with an individual who's passed away and it is on a flight, as opposed to like, we have it downstairs in our hospital suite. So like, all of these things add variants that we can't really control. And then again, we're adding the variants if we're doing it over the course of years. And we're doing different lots for antibodies and the instruments getting older, things of that nature. And so the first thing is to accept that there's lots of variants and then smile at it, because you're not going to reduce all of it. And so it's important to think critically of how you actually handle it appropriately. So one of the things that I developed that I didn't actually get a chance to talk about is incorporating standards onto the tissue for measuring and instrument performance as a function of time. And so we can actually monitor the instrument's performance prior to touching the sample. So if there's an issue, we can either fix it or we can use it to actually like normalize our data. And these standards are synthetic. They have a different mass deviation than our biological samples, so they're easy to pick up. They function very similar. We've measured RSD and all that stuff, and they actually do a really good job measuring instrument performance. And we have this little GUI that I built with Tina that will tell you like, oh, this is indicative of the slide not being flat, and it's in fact like rotated cockeye this direction. And so using that, it really allows us to measure instrument performance, allows us to monitor laser fluence, because the laser will eventually die over time. They last about 5 billion shots, which is good, but gets you about a year when you're using it constantly. For antibodies, they were actually banned by the NIH for a while because they were too variable. And so I use recombinant so that we remove the animal aspect. So I use antibodies produced by bacteria, so they're much more reproducible than they have been traditionally. And then we use machine learning. And so using machine learning and saying, I want the computer to tell me the best differences between our population of men and women, it's less likely to tell you an artifact is the main discriminator unless men were treated in one way and women were treated the other. Those will usually not have high discrimination values. And so I randomized everything from sample collection. Inherently, people have surgeries on different days, so that helped. But like everything was randomized such that not all women or not all highly obese patients were analyzed on the same day.
Great. And thinking about all that data that comes out of any given project, how developed are algorithms that sew together MALDI tessellations? Is that like generally an area that needs development, or has it been solved primarily? I mean, you spoke a lot about machine learning and things like that. So could you speak to that?
Absolutely. So we need to come farther. A lot of this is custom. Now I do cheat, and I'm not ashamed of that. So I borrow things from other communities. Spectroscopists and fluorescence microscopists have done high resolution imaging and like image registration and stitching and stuff and background subtraction for 100 years now. So they have all of this figured out. And so one way I do this is instead of making everything custom, I take what they've started with and just bend my mass spectra into the format they want. And that helps a lot. I do that with cellular neighborhood analysis. So I do molecular neighborhood analysis based on how microscopists do cellular neighborhood analysis. And so a lot of the machine learning was custom, but I think there's a lot you can actually borrow from other communities. And I think we need to do a better job. My field should do a better job borrowing instead of reinventing.
Oh yeah, definitely. I mean, like even in metabolomics, if you don't tell something is, a spectra or whatever it is, does it really know? You just can feed it some values and it might work. So definitely, yeah, I promote the borrowing as well. Jumping to the next question, without eye mobility, the image data could be biased by interfering high signal matrix isobars without one knowing. How do you see a promising way to reduce or at least monitor such biases?
Absolutely. So I will say the higher your M over Z value, the less your matrix interferes. It doesn't cluster that high, especially when there's other molecules, and we can discuss why offline. But for small molecules, it's a huge problem. And so that's why I really think there's two ways to really handle that. One is just use eye mobility. I have eye mobility downstairs. There's a reason that's the instrument I bought, because I just like, why punish yourself? Two, you can actually handle it somewhat in the mass resolution range or mass resolution, because matrix often have different mass defects than your small metabolites do. And so that's a way to kind of cheat it out. So as long as the resolution on your instrument is better than like is pretty good, you can cheat that way. Other alternatives include like derivatizing your small metabolites to actually push them at a higher M over Z value so that you're not fighting the metabolite range. But eye mobility is the fastest and I think the easiest at this point, but all of them have merit, and there's definitely reasons to try them all, actually, depending on what you need.
Is that a general rule for like all matrices in terms of like not mixing so much with higher M over Z ions?
Largely, there's some exceptions to this where they actually, so people have designed matrices that actually are in the higher mass range,
Okay.
so that they're not they're not usually isobaric in the small metabolite range. There's pros and cons to those. They often don't crystallize very well, because if you think about crystallization, you have to like pack these things together and it has to crystallize, and the larger the molecule, the less packing you get often. So the crystal size is rough, so you're like killing your spatial resolution.
Okay.
So I think really there's a lot of different options, and it's important to just know what you need from an experiment. If you could care less if your spatial resolution is 100 microns, then yeah, use those with like inherently no background in the small molecular space. But if you really need high resolution, then eye mobility is probably better, but you're also adding 50 milliseconds per pixel, so it gets much slower. It's about an order of magnitude or two slower. So you pick and choose your poison.
Yeah, definitely. Can you tune or optimize the ion mobility for specific lipid classes to get better separation within that class, or is one general TIM setting the best?
You definitely. So it's important to optimize, and this is what I've tried to instill in everyone I've ever mentored, is like, you need to optimize. So usually how I think about the TIM system is I have like a couple general methods where like, I know that this will work for small metabolites in general. Like I will get separation and it will be fine, and I transmit ions. And you do that for like peptides and lipids and all that stuff. But once you have an idea of what things you want to separate, you can start actually dialing in and say, I want to separate like stereoisomers of lipids. You can actually optimize your separation of these two lipids. It just takes time. But even like, I know Jonathan Sweetler on his system is separating RNS or D-amino acid containing peptides from non-D-amino acid containing peptides. And that I helped them optimize it. This was actually quite a bit ago, and then they've actually got an imaging system now. But you can absolutely optimize to get better separation depending on how much time you're willing to sit there, and the experiment gets much longer, so it's not practical to do like these small separation windows for like an entire image.
Great. Yeah, that's some great feedback. Moving on, curious that you have to look into correlation between protein markers with metabolites. Why is that?
Can you repeat the question one more time? Sorry.
So it was more of like a comment question.
Okay.
Curious that you have to look into correlations between protein markers with metabolites. Why is that?
So I showed let me take a step back. I am picky with how I word things. So I showed proteins, but those proteins correlate to a specific cell type in the kidney. So I'm saying like nestin, but it's really nestin stains a specific cell type in glomeruli. So I use those interchangeably, but you are actually detecting the protein. And so why I think that's critical is that we know a lot about transcriptomics in our system. Like that's how we understand most of biology. Now it's great and powerful and cool, and there's lots of reasons transcriptomics is neat, but it doesn't always correlate with proteins. And that's like a direct connection, right? Like you express a gene and you should probably get the protein. And we know that that's not true in a real system. Similarly, we have proteins, and we know those proteins interact with metabolites. And you would imagine a world where those should be correlated, but they aren't always. And so there's twofold reason that this is important. One is we need to actually measure, is there actually like correlation between these two things that should or should not be correlated? And two, we actually don't know the function of many of these like lipids. And so like that oxidized phosphatidylcholine, we don't know what it does. And so by finding out that it's highly localized to the collecting duct, and if we can take it a step further in different cells in the collecting duct, we can start maybe piecing together what this specific lipid does. And that's something that I think a lot of metabolomics has doesn't necessarily know, especially in the lipid space, what these things are actually doing.
Okay. I hope that answered that question. And you just mentioned oxidized phosphatidylcholines. There's so many questions on that, and we can maybe try to go over a few of them. So I heard that MALDI MS imaging suffers conversion in source from PCs to PA. Do the lipids that you identified as PAs, do you think they're actually PCs?
So this is a great question. And the answer is it's super important to validate what you're looking at. So they could be. So what we've done, and I didn't talk about this, but this conversion in the MALDI source is matrix dependent and pressure dependent. And so you can actually change the pressure and actually see if you're converting or if you're not. You can also change the matrix to see if you're converting or you're not. So there's matrices that maybe can't get high spatial resolution, but don't convert because they're very low energy. Like the plume is just lower energy. So you can actually go through and detect and say, oh, is this actually a PC or is it a PA? And again, it's not perfect because it's not the same tissue, but it gives you some evidence. We also do 10 MMS and orthogonal LC-MS experiments. So I'm not claiming anything in the MALDI that isn't also in the LC. But there's we're still figuring out that if there's any conversion, but absolutely there is conversion in MALDI, and that's something I'm aware of. So we try to like provide evidence for or like orthogonal validation of some of our species.
Yeah, you did mention that. Another question related to PCs and PAs, as PA can be a fragment artifact of PCs, kind of similar to what we just talked about. Would you think the PA is derived from laser in-source fragmentation or during sample prep? For example, if you were to use a PLD enzymatic cleave.
It depends. So the most that I have seen, and this I will admit is probably sample prep, or not sample, it's sample specific and instrument parameter specific. But you mostly get the like fragment artifacts in source, and it's a function of your pressure and your matrix. So this is a lot, and we can talk offline, but basically your matrix absorbs different amounts of laser energy. Some are better absorbers than others, and so because of that, there's different energies. There's like hot matrices, which are high energy, and that causes a ton of fragmentation. There's some that are more stable and actually don't absorb that much laser fluid, so it's like fairly soft, and those don't produce as many fragments. So it's hard to give like a blanket statement, besides it's probably both, but I've encountered the in-source fragmentation from like just the matrix choice. So like DAN, I like DAN, but it fragments everything. It's very high energy.
And DAN stands for?
D-amino acid something.
Okay.
I should know.
No,
I just
okay.
call it DANs, and it's got numbers. Let me look.
Oh, gotcha. No, it's fine. I wasn't sure if that was a person or not.
1,5 D-aminoethylene. I'm in a
Gotcha.
chemistry department, and I actually don't do proper chemistry.
It's okay. School doesn't start until the 29th. So
That's
one last question, and I think it's an important one. Is there a standard or accepted number of biological replicates needed within the MSI community? What do you normally do? What's routine? Can technical replicates of serial sections be used to validate findings instead of having more animals?
Yeah. So this is a lot, right? So
Yeah.
there's lots of different ways to answer this. And like, it's a very highly debated topic. If you're ever interested, you can go to ASMS and our imaging subgroup, whatever, and you'll see people yell about it all the time. There's two things that are highly debated. One is the definition of spatial resolution. That's highly debated. And the second one is like, is there even technical replicates in MALDI? And I mean, honestly, if you think about it practically, there isn't a technical replicate in MALDI unless you image the same tissue, but even the function of imaging a tissue twice changes the tissue. So
Yeah.
like, this gets into like, how do you actually monitor, do like, it's very different than LC-based approaches where you can make a technical replicate. So that's why I use like standards and things, because those are things you can actually like very reproducibly apply. And
Mhm.
there's like, I'm exploring tissue mimetics where like, can we homogenize a tissue? Can we then make that where it's more like a technical replicate by homogenizing it and spotting it or whatever? No size fits all. So there's different ways to think about how many samples you need. So I have done like, I mean, for grants, you have to explain how many animals you need and whatever. So for medium abundance ions, it's 4 samples per demographic that you're trying to assess. So say I want to know the difference between cancer and non-cancer, I need 4 cancer, 4 non-cancer. For medium-sized ions. For smaller ions, you need about 6 to 7 is what the math shows, more or less. And then I think going beyond below 4 is a bad idea, even if the math tells you that you can do 3. Now, clearly, the better the N, the more appropriate you're going to be able to model what the population looks like. And so I think the answer is 4, and then you add more as you're able to. And sometimes you're not going to be able to get many samples. So like in the case of autism, I can get more control samples than I can with individuals with autism. So if you better define your healthy like population, that allows you to reduce your number of N on your like disease state, because the better you've modeled your healthy population, the better you can kind of assess how this smaller population is different. So there's different ways to cheat.
Those are some great tips. I mean, especially for those starting out, or, you know, just with recently who've acquired new instruments. So yeah, with that, we've come to an end. If there aren't any last questions or whatnot, I'd like to talk about our next speaker for next month. Thank you, Elizabeth.
Yes, thank you so much.
Thanks for the opportunity. I appreciate it.
Good luck with the quarter.
Thank you. I have to pretend to be a chemist. I'll actually start heading to y'all's. Should I just meet at the building?
Yeah, we'll be here.
Sweet. I will go head that direction.
Okay, so next month we have Dr. Evie Mathey from the NIH NCATS. She's the director of informatics, where she leads a diverse team of experts in bioinformatics, cheminformatics, you name it, data science, software development. We're very excited to have her. Her lab is currently developing computational resources, methods and tools to optimize not only large-scale molecular projects such as like high throughput screening or multiomics, but also knowledge-driven data sets, which just means like looking at various information such as like information on drugs or including mechanisms of actions. So we look forward to hearing about her robust data analysis frameworks for multiomics data sets. Please join us in October. With that, thank you. Thank you for joining us today.
Thank you. See you soon. Bye-bye.
Bye.